DNA and mutations. Before we get into mutations, we need to know what DNA is. DNA, which stands for deoxyribonucleic acid, is a molecule that is made up of two polynucleotide chains that coil around each other to form a double helix. It carries the genetic instructions for development, functioning, growth, and reproduction of the organism. The structure of DNA is composed of building blocks called nucleotides. Each nucleotide consists of one sugar molecule called deoxyribose, one phosphate group, and one of the four bases adenine A, guanine G, thymine T, or cytosine C. There are two types of bases, purines and pyrimidines. Adenine and guanine are purines as they have two rings, while thymine, cytosine, and uracil are pyrimidines because they have one ring. In the image over on the right shows the double helix phosphate backbone and the bases in between the phosphate helix. Urin-Chagraff's rule of base composition of DNA shows that the amount of A equals the amount of T, and the amount of G equals the amount of C, or the amount of adenine equals the amount of thymine, and the amount of guanine equals the amount of cytosine. So adenine forms a hydrogen bond with the thymine, and guanine forms a hydrogen bond with cytosine. So in the picture, we can see that the adenine bonds with the thymine, and cytosine bonds with the guanine. Now, a section of DNA is a sequence of bases codes for a specific trait of an organism, which is the gene. Now, RNA is a nucleic acid that carries genetic information from the nucleus to the cytoplasm. Unlike DNA, it is single-stranded, and instead of thymine, it has a uracil base. There are three types of RNA. Number one, messenger mRNA, which complementary copies DNA almost identically and carries protein building instructions. Number two, transfer RNA, tRNA, which delivers amino acids one at a time in a ribosome. And finally, number three, ribosomal RNA, rRNA, which is a component of the ribosome. Just like we discussed, the messenger, which is the mRNA, looks like this image on the right. There are the bases uracil, adenine, guanine, and cytosine, but only one strand of it. RNA can go out of the nucleus because it is small enough to go through, unlike DNA. Then we have transfer RNA, which is tRNA. It is easy to remember because it looks like a lowercase t. It delivers amino acids one at a time into a ribosome. Finally, we have a ribosomal RNA, which is rRNA. It is the component of the ribosome. The sequence of nucleotides of DNA is transferred into a sequence of amino acids in a protein. There are two steps to protein or protein synthesis, or aka central dogma. Number one, transcription. Bases in DNA transcribe into bases in mRNA. Number 2, translation bases in mRNA translate into amino acids. And to proteins that are made, there are four structures. Primary structure, secondary structure, tertiary structure, and quaternary structure. Number 1, a primary structure is the sequence of amino acids and determines everything else about the final product of the protein. It is the top part of the image on the right. Secondary structure is the structural motics which include the corkscrew-like alpha helix, beta pleated sheets, and less organized random coils, which are part of many polypeptide chains. Tertiary structure are motifs that are in a large scale turn. Quaternary structure is several polypeptide chains that are linked together in the protein. Now that we learned what DNA is, what is mutations? A mutation is a permanent change in a cell's DNA base sequence. If the mutation occurs in the egg or sperm cell, it is passed down to the next generation. There are 3 types of mutations: substitution, deletion, and insertion. Substitution mutation is when a single base is substituted for another in a codon. It may or may not affect the amino acid or protein. Let's see how this would work as words with 3 letters in them. The sentence, "The cat ate the rat," would change to "The cat ate the hat" if we substituted the letter H in for R. We can see that the sentence still makes sense, so if this was a codon, the amino acid or protein would not be affected. However, if we were to change the letter A in ate with a P, the sentence does not make sense, so if it was done in a codon, the amino acid or protein would be affected. However, the effect can be minor or major depending on where and which base is substituted in. Deletion mutation is when a base is deleted or removed from the sequence. It causes the triplet frames to shift. It always affects the amino acids and the protein. Again, the example of "the cat ate the rat." If we deleted the letter A, the sentence wouldn't make sense as the codon shifted up to regroup as a group of 3. We can also see that one of the groups are not a group of 3 and instead it is a group of 2. Again, the effect can either be minor or major depending on where and which base is deleted in, but there is still an effect. Finally, insertion is when a base is inserted or added to the sequence. It causes the triplet frames to shift. Like deletion, it always has an effect on the amino acids in the protein. Using the sentence, "The cat ate the rat" again, if we inserted the letter D between H and E, the word "the" in the word the sentence would not make sense and the codon is shifted down to regroup as a group of 3. Now, mutations aren't all 100% genetics, and there are many possible mutagens, such as radiation, toxins, viruses, and errors in DNA replication. A great example of mutations are cancer. There are many types of cancer, but it is an abnormally growing and dividing cells of a tumor. Malignant masses of cells that lost control over how they grow and divide. It disrupts the surrounding tissues both physically and metabolically by hogging up the energy and nutrients that are given to the group of cells. The cytoplasm and plasma membrane of the cancer cells become grossly altered. Cancer cells have a weakened capacity for adhesion, and if left untreated, it usually has lethal effects. That's all for today's lesson. Hope you learned a lot and see you next time.